morning, everyone. Nice to see people here bright and early in the morning. My name is Aditya Ramdas. I'm a faculty at CMU. You'll hear from me later on today. I'm just here to introduce our first excellent speaker for the day, Caroline Müller. Caroline's a faculty at MIT, and she's known in a wide variety of fields for her work, from computer science to statistics to mathematics to philosophy because of her interest in causal inference. And she's contributed to a huge number of topics, and she's also an excellent speaker and a good friend. And so it's my pleasure to introduce Caroline.
Okay, thank you very much for the invitation to be here. It's a pleasure. It's my first time really visiting Mumbai. Otherwise, before I was only flying through, and I've been really enjoying it. So thanks a lot. What I decided I wanted to talk about is different topics around graphical models. So the first lecture, I thought I will concentrate more on the directed setting, on like how can we infer causal relationships from data and how can we represent them by graphs, so by networks. And then the second part will be on somehow the opposite, and you'll see how it is the opposite, namely something known as total positivity, which can be used for modeling positive dependence among data. And that's really very, we'll see that actually whenever you have total positivity, that's really the opposite of a causal structure. And we'll see how that actually really comes in when we actually look at graphical models. But just to give some more motivation, starting from applications. So what are these graphical models? So probably many of you are familiar with graphical models. So graphical models are some probabilistic models. So you have your random variables, and you represent the statistical relationships among these variables via network. Now, this network can be directed or it can be undirected. It can have latent variables. It cannot have latent variables, etc. And depending on the application, you'll want to use undirected graphs or directed graphs to represent these relationships. For example, if you're thinking about weather forecasting here, undirected graphical models are used a lot because you cannot intervene on the weather, right? So it doesn't even make sense to think about something causal when you're thinking about the weather, because I cannot intervene on the weather in, say, Las Vegas and then see how it acts or how it changes the weather in LA. But you do want to know something about the relationships among the weather here because, right, so for example, if you have like big mountain ranges, then you might have, you know, effects or correlations, strong correlations on one side or on the other side of the mountain range, but, you know, very little across the mountain ranges, right? And these are used in order to actually do accurate weather forecasting. Once you know all these correlation structure, that, of course, then can tell you something about how the weather will be affected in the next days in a different place. So that's where undirected graphical models are used a lot. Now, if you go to other applications in medicine or in biology, there you really care about causal relationships, right? So if I want to develop a new drug, I need to know what happens if I intervene on a particular node, right? I want to know the different targets, and now if I intervene there, then only something downstream is going to change, but nothing upstream is going to change. So here I really care about the causal relationships, and in fact, I can intervene on the system, so this is where you want to be using directed graphs. Okay, so I'll be talking about, in the first lecture, I'll be talking about something that is in the directed setting, in the second lecture, something in the undirected setting. Okay. So just a bit of history about graphical models. So what is the motivation? What is the motivation for actually using these graphs to represent joint distributions? Well, it's really to make the distribution, to not have to look at all possible joint distributions if you have, say, 10,000 variables, but to really only look at simpler distributions where you can write it in an economic way to get interpretability, first of all, but also often these graphical model structure, at least when it is sparse or it doesn't have high degrees, then that allows you to do pretty efficient inference, or we'll talk about how efficient these kinds of things are. So that's really the motivation for graphical models, and it's a really nice kind of whole theory that combines graph theory together with probability theory into a pretty powerful framework for actually looking at multivariate distributions. And in terms of history, it's really three different areas that came together to build the foundations for graphical models. That's on one side, Ising models, right? That's in the undirected setting, coming from statistical physics. So in the discrete setting and undirected graphical models. That's through Gibbs. Then where the directed setting actually came in pretty much at the same time, maybe a bit later, is, and I'll talk a bit more about that, is through Sewell Wright. He was studying inheritance. And by studying inheritance, he actually laid the foundations for directed graphical models and causal inference. And then also pretty much at the same time in statistics, people were studying how to represent interactions. They were studying contingency tables, so count data, and interactions among these variables in contingency tables. So again, in the discrete setting, and looking at graphical models there. So all these came somehow together to form the basis of graphical models in the undirected setting, discrete setting, continuous setting, and then also in the directed setting. Okay, so that was a bit of the history. And so what I will concentrate on is really in this lecture here in the directed setting. So as I said, so the first lecture today will be about causal inference. And what do I want to do? So as I said, this is a very old area, right? So Sewell Wright actually laid down the foundations already in the 1920s. But everything until very recently was all about observational data. So if I just have observations, how can I go from observations to something that is causal? But now what I think is really exciting and why I'm really excited about thinking about causality is that for the first time actually in various different applications, we have access to interventional data. And I'm particularly interested in genomics, but you can also think about advertisement industry, etc. Right? For the first time, you actually have really large-scale interventions. Like, for example, we can do gene knockout experiments, right? You can take, so humans have 20,000 genes. You can take any one of your genes and you can knock it out. So you can set the expression to 0, right? And you can see what happens on all the other genes. Same, you can do personalized ads, right? Very, very easily nowadays. You can do drug interventions. There are nowadays these huge drug testing platforms, right? Where you throw small molecules at your cells and you take images from it. So there is really a large amount of interventional data that is becoming available. And so I think what is really needed is that we start thinking about how can we use interventions. Right? Interventions are not anymore only here to test causal relationships that you get from observational data, but you want to be using interventional data already at the beginning in order to learn the causal relationships. So we need a new framework for thinking about observations and interventions together in order to come up with causal relationships. And so here, how we're thinking about this, so what does the data look like in genomics, for example? So these are the random variables. These are genes, so humans have 20,000. And then these are the observations. So actually nowadays everything is measured in single cells. So these are your IID observations here. And these observations, usually the data sets nowadays are like 100,000 to 1 million single cell observations. So that's how many samples you have. And as I said, you cannot just have observations, but I can also intervene on a particular variable, set one of the variables, say, to 0, and see how all of the other genes, the expression of all the other genes changes. Okay, so here, for example, I would take, gene 1 and set it to 0, then I take gene 2 and set it to 0, then I take gene 1 and 10 and set it to 0, etc. So you can do any of these interventions. If you do too many at the same time, the cell will die. Okay, so it's not that you can intervene on 100 nodes at the same time and then you just get a dead cell, so that doesn't tell you much about it. But still, the space is huge, right? So 20,000 and, you can do combinations of interventions as well. So that's the kind of data that is available in many different areas nowadays. And so the question is, how can we use this in order to do causal inference? Okay, so that's what I want to talk about now in this lecture. And then just to give an overview on the second lecture, and why is that actually the opposite of causal inference? So I think in many different, and again, my motivation again comes from genomics, and I'll talk a little bit about it, but let's maybe more people are familiar with finance applications. So you'll often see these kinds of curves when you look at stocks, where you see that, stocks, okay, some of them move a bit more and others move a bit less, but many of them will be moving up and down together, right? They'll move a bit more up or a bit less up, but they will be having all these trends where they're moving together. So there's very strong positive dependence, which is introduced because there is some latent variable, right? Which is some market variable, which introduces this positive dependence. Now, when you see something like this, there is a causal structure, namely this latent variable is causing everything to be moving up and down. But if I just want to model the observed variables, there is no causal relationship between them, right? So everything, they're moving together just because there is this latent variable, but there is really no causal relationship between them. So this is really the opposite of causal inference, right? So here I will have undirected graphs that represent these kinds of movements. And similarly, I mean, we're thinking about gene expression. So what happens actually in the cells, so here, you know, we have all these different cell types, right? We have skin cells, we have neurons, we have liver cells, et cetera, et cetera. They all have very different expressions, right? They look completely different if you look at a neuron or if you look at a skin cell. And what actually makes them different, or in part what makes them different, is the packing of the DNA. So the genetic material inside the cell nucleus. Because if you pack these genes differently, different genes can come close in space, and hence, you know, they can interact, right? These genes here now interact. And so something comes in and turns on all of these genes at the same time. And so if I repack it differently, then other genes will be turned on at the same time, right? So the co-expression pattern will change completely. But this is, again, something that is not causal, right? They're just together in the same place, and hence they're all turned on together. But there's no causal structure among these three genes that are close in space. But they will be hugely positively dependent, right? They will all go up or down together, but there's no causal structure among them. So how we're thinking about it in terms of gene regulation is really, there is a causal structure, you know, the proteins that come in and then cause all of these to go up. So there is a causal network between these local clusters. But within the local clusters, it's not causality, right? It's something that is this positive dependence that we have to think about how to model it. And so that's what I want to talk about in the second part. So how does one model strong forms of positive dependence in data? And what are consequences for graphical models? Okay. And we'll in fact see that whenever you have positive dependence, you cannot have a causal structure. So it is really the opposite of that. Okay, so that's what I would like to do in the second lecture. Okay, but first we'll talk about actually this causality and how do we use interventional data in order to learn causal structures. Okay, so I will be really using this very old framework by Sewell Wright that I mentioned. That was developed in the 1920s, so that's the framework of structural equation models. It's a very intuitive framework for thinking about causality, or he introduced it to think about inheritance. Right? We have all of our random variables. For me, they are genes, but they can be any other random variables as well. So these are these x's, these are random variables, and then I represent the causal structure by a directed graph. Okay? Now, because it's causality, right, I cannot cause anything that happened behind me in time. So this will always be a directed acyclic graph. Right? There cannot be any cycles. And this was introduced. Nowadays, there is generalizations to also having cycles, but of course, when he was thinking about heritability, there were no cycles, okay? So I will be using this first framework that was introduced, namely that it is a directed acyclic graph. Yes?
In the level of data structures, will you be storing Fs on the conditional?
Yeah, so let's talk about that maybe afterwards. So for me, in this talk, all I'm going to be wanting to do is actually learn this graph. Okay, so I will actually not talk about learning these functions. I'll just be talking about learning the graph. So usually one first learns the graph, and then afterwards one does either standard linear regression if the functions are linear, or nonlinear regression, etc. So that's somehow, I will call it an easier problem than the first problem of actually learning the graph. So I won't talk about it at all, but there are all kinds of problems there as well. Okay. So now you're already getting to my model. So what is the model? So now we have a graph which represents the causal relationships. And I want to keep it as general as possible. So I'm going to be working completely in the non-parametric setting. So every variable here is just a function of its direct parent and some noise. So all these epsilons are independent noise variables. And so, for example, x4 is some function of its direct parents, x2 and x3, and noise. Okay, completely non-parametric. I'm not making assumptions that epsilon has to be Gaussian. I'm not making assumptions that these f's have to be linear. They can be anything, right? And that's super important for me in genomics, right? They're not linear and they're not Gaussian noise. Okay, so this is the framework that I'll be using. And what I want to do is from data, right? From data on these x's or interventions, I would like to learn this graph. I'm not going to be talking about learning the f's. Okay, so this is the framework that was introduced by Sewell Wright already. Okay, so this has just observational data. So if I would just have observations here, I would like to learn this. This is very well understood. Now, what is new is that we actually have a whole lot of interventional data. Okay, so now we need to figure out a framework for how do we think about interventions. So first, okay, from observational data, you know, we had like a huge, so in the 1920s, the framework was set out by Sewell Wright. It was basically forgotten by statisticians. There was a big fight about how can you go from observations to causal structure. And then really it came up again through very large developments by Julia Pearl, Jamie Robbins, Don Rubin, and Peter Spertus, all about 1970s, 80s, 90s, etc. Okay, so that's all in the observational setting. Now, what I think is really exciting is that we actually have a whole lot of interventional data. And I'll be distinguishing two different kinds of interventions, again, motivated by genomics applications. But I think they really encompass basically all interventions that you can think about. So the hard interventions, those are the most invasive interventions. So what are they? So I just take some of these nodes. I can actually also take multiple nodes. But say here I take one node, x2. And I'm deterministically setting it to, or not, it can also be random, but all I'm doing is I'm removing the whole dependence of all of its parents. Okay, so x2, so in the simplest setting, let me just do a knockout experiment, and I'm setting x2 to be equal to 0. Okay, when you set x2 to be equal to 0, there's no more dependence on x1, right? I don't care anymore what x1 does. x2 is just be set to 0. So I'm changing the graph structure when I'm doing a hard intervention. I can also set it to some random noise, right? I'm still changing the graph structure when I'm doing that. So that's a hard intervention. When I'm changing the graph structure, I'm removing all of the effect of the parents onto me when I'm intervened on. Okay, that's a hard intervention. Everything else is a soft intervention. Okay, so in a soft intervention, I still have the effect of the parents. I can either change the noise or I can change the function. Okay, so that's the difference between the two. So this in genomics would be, for example, a gene knockdown experiment. Right? I'm removing some of the protein or some of the RNA that is being produced, but I'm not completely changing the structure of, you know, how someone before, how my parents acted on me. Okay? So I will be distinguishing these two, and now we can of course ask, well, what are the questions that I'll want to ask? And so let me do that on the next slide. But first, let's just look at what was known, and it's very well known in the observational setting, is if I just have observations, then I cannot uniquely identify the causal structure, right? So if you just have observational data, so this is known as Markov equivalence classes, if I just have observational data, I cannot tell if X causes Y or Y causes X, right? They're just correlated. Okay, so these are the Markov, so on 3 nodes, we know that just from observational data, I can identify these different equivalence classes. So for example, if you have a structure like this, where Z and Y causes X, I can actually identify it uniquely because this is the setting where I have X is independent of Y. Okay, in no other setting will I have X independent of Y. Okay, so this graph here, if you look at its conditional independence relations that are implied, it will uniquely identify this kind of graph. But in all other settings, right, the conditional independence relations that you get can identify multiple ones of these graphs. So they're just the same for multiple graphs. And this is very well known. It's exactly, you know, there is even so you don't have to look at the probability distributions and their factorization, but you can actually just look at the graph. And it is well known when can you actually identify the graph or which graphs are equivalent in the observational setting. Namely, they have to have the same skeleton and immoralities. And what are these immoralities? Where there are these, you know, these V structures. So where you have 2 nodes pointing to another node. So it's known as an immorality because it's 2 parents that have a kid, but they're not married. Okay, so here you see like philosophy, etc., entering in, right? In terms of like how you name these kinds of things. So we'll have things like faithfulness, etc., coming up later on. But that's how you, okay, so this is an immorality because these 2 have a kid, but they're not married. So there's no edge between them. Okay, so there is a very nice graphical kind of criterion which tells you, you know, which graphs can you actually identify from observational data or which ones are just equivalent in terms of observational data. Okay, so this is very well known, right? This is like 1990s. And so what are the kinds of questions that I now want to ask? Well, now we also have interventional data. So hopefully when you have interventional data, right, then these classes will hopefully break down more, right? You hope that you can actually identify more of these graphs. In particular here, right, if I intervene on node X, then in this case, Y changes, but here Y doesn't change. So I should be able to distinguish the two. So it is definitely, it should be the case that, when I have interventions, the Markov equivalence classes become smaller, and the question is how much smaller, and can we really, write down a nice graphical criterion for this? Also, what I'd like to know, and this is something that came up from genomics, is there actually a difference between hard and soft interventions? So people were actually doing always hard interventions because they thought gene knockouts and not gene knockdowns, because it was thought that that provides you more information about the gene regulatory network. Okay, these are more invasive interventions, so hopefully they will provide you more information. And so that's a question that we wanted to ask. Is it actually true? And it turns out it's not true. Okay. And then, of course, once you know what you can identify, then you can start coming up with algorithms that can actually identify this, at least as the sample size goes to infinity, right? So as long as you don't know what you can identify, you cannot come up with provably consistent algorithms. So only once I know this, then I can start actually coming up with algorithms. So this is what I'd like to do now. Okay, so first, what are these interventional equivalence classes, and how do they depend on the type of intervention? Okay, so this was interesting from a genomics perspective. So first of all, so this is the main theorem. So these interventional Markov equivalence classes under hard and soft interventions are actually the same. Okay, so you do not get anything more by doing hard interventions as compared to soft interventions. So that's very interesting from a biological perspective, because when you do, as I said, when you do hard interventions, usually the cell dies when you do 5 or more interventions. When you do soft interventions, that's not the case. Okay, if I don't perturb the gene too much, right, but I still perturb it, I can actually do 10 interventions in the same cell and it doesn't die. So that allows me to actually test, much higher order interventions, which is interesting from the perspective of obtaining data. And we also know exactly what can be identified and what cannot be identified. So basically, everything, of course, that was already orientable from observational data is still orientable, because I do have observational data available. And then in addition, it's kind of the worst case, so you would hope that maybe something more can be identified, but it's just that you can identify everything that is neighboring to the intervened node. Okay, so if I go back here, so if I would have an intervention on node X, then I can distinguish these two guys. But let's look at this class here. If I have an intervention on node X, I can still not distinguish this guy and this guy, right? Because both of them, I can now tell that the edge points in, but I can still not tell for this edge if it points in or out. But I can distinguish this middle guy from these two. Okay, so here the equivalence class would have one more guy and then these two guys together. Okay, so that's a nice kind of graphical criterion. So for hard interventions, this criterion was done in Hauser and Bühlmann, and we showed that it's actually the same also for soft interventions, that you're not losing anything even when you have soft interventions. And what we just recently showed is actually that you don't even need to know the intervention targets. You can identify the intervention targets. Question is, of course, how do you do that computationally efficiently? I'll talk about this briefly, but you again get exactly the same kind of result.
I guess if you know a little bit more about your F, then you can
Perfect.
identify more than this.
Very good, yes. In fact, so this is very interesting that if I go back, so I can either know more about my Fs or know more about my noise. So if you know that your noise is strictly non-Gaussian, then you can actually always identify each one of these graphs. If you know that the relationships are strictly nonlinear, you can identify all of these graphs. Now, the problem is always that you always have this problem of like statistical and computational properties, right? So you need to be quite far away from Gaussianity when you have even like 10,000 or 100,000 samples in order to really be able to identify these different kinds of graphs. But theoretically, yes, it helps you greatly. Okay, but now with, for example, nonlinearity or non-Gaussianity, you're paying a huge price in terms of computation time. Yes, but it's true. If I have additional assumptions and I have huge amounts of samples, then yes, you can actually use it. Yeah.
In the case of a hard intervention, I know what I have done in the sense that it's a gene knockout. But in the case of a soft intervention, is it enough to know that I have intervened, or do
Correct.
I know the nature of it?
No, you just need to know that you have intervened. And
in a biological setting, we have lots of sort of causal cycles in the sense that if I unroll it in time, then of course it's
perfect.
And
Yes.
so how do you deal with it?
Yes, exactly. So, okay. So, so there are two answers. So one is either you go the more difficult way and you actually assume that it's not a DAG, and then you pay a huge computational price. Or, and this is how we're thinking about it more, is, you know, if I give a biologist a DAG like this is obviously not looking like a real gene regulatory network. Okay, so what it means for me, or how I define to understand a gene regulatory network, is if I can predict the effect of any intervention. That to me is good enough in terms of understanding, right? If you have never if you have never knocked out gene 10, and I'm from this data able to tell you what happens when you knock out gene 10, then people are usually happy with it. They don't actually care about these gene regulatory network. So it's kind of the same thing. It's like when you do linear regression or graphical lasso, right? I mean, all I want to be able to do is do good prediction. As long as I have a representation of this causal network that allows me to do good prediction of an unseen intervention, I'm happy. So I'm just seeing this causal representation as a representation of the network or of the causal structure that allows me to predict how well I'm doing when I'm intervening some other node. So I'm not seeing this network as a true network. Okay. And this is also when we actually evaluate how the method does. How we do it is we always leave out one intervention and we try to predict the effect of it. That's how we draw ROC curves, etc. So that's anyways how we're always evaluating these things. Because we don't know the true network structure anyway, so this is the way to evaluate it.
Do you need to assume that F2 tilde is not F2 or epsilon?
Correct,
tilde
yeah.
Epsilon 2, because otherwise...
These have to be different than these, right? Yes. So there needs to be an effect of the intervention. But this you can even see in the data, right? I can see that I'm actually changing something in the distribution. So all I need is that I'm changing the distribution of X2.
So it's just the F2 increase. You don't care about epsilon 2.
Either one of the two. I can either change this one or this one. Yeah, doesn't matter.
For the identifiability question.
For the identifiability question, yes, it doesn't matter, no. And you know, because it's infinite data, it also doesn't matter how much I'm changing it. Yeah. So all of these questions come up when you have finite sample size. Okay. So that's for the identifiability question. Okay. So now that we know what we can identify, now the next question is how do we identify it, right? Can we come up with algorithms that actually give us these kinds of Markov equivalence classes? And I mean, finite sample questions, there are many more. So I would think that so here we're saying that, okay, so you get the same identifiability results, but you're actually paying a price in terms of computation time. So how we get this is, you know, if you're thinking about the Gaussian setting, for example, then in the hard intervention setting, you just have to do conditional independence tests. Now in soft interventions, what we need to do and how we get this result is by replacing conditional independence tests by invariance tests of distributions. So what I need to, I'm checking whether, you know, the interventional distribution and the observational distribution, whether they're shifted or not. Right? So these are harder, I mean, there is no hardness result, but the tests we're using require much more computational time than the conditional independence tests. So I would believe that one can prove this, right? That this is computationally harder to do. But I think that when you have finite sample, I mean, there is also something, and this is like using the strength, right? So using the strength of the intervention, I think there is something to be gained statistically, namely that it's easier for me to estimate the effects, the causal effects, right? But there is no theory such. But I think there is here a very nice statistical computational trade-off where if you have soft intervention, you know, you're paying a price in terms of computation time, but you should be gaining in terms of statistical properties when you want to estimate the causal effect. That's just a hypothesis. Yeah.
I'm a bit confused still. So suppose you have a node and you intervene on it and you determine that there is an edge to some other node.
Correct.
Doesn't that give you a way to intervene on that other node as well?
Oh, yeah.
So that you can use that one to identify the edge to a third.
Perfect. Yeah, yeah. Okay, so let me actually say this. So I didn't write this down, but like, so yes, you can identify the nodes that are adjacent to an intervene node, and this might identify some other nodes down the street. That's correct. Yes. So, but this will tell you exactly which ones. Okay? But in particular, it's exactly what you're saying. If I have a path outwards, then I can identify all of them. Because I know, because from the observational setting already, you know that I can identify when there is a V structure. So V structures are crossing what you're just saying, right? Whenever I have a V structure, there is no more effect downstream, and then I cannot identify anything anymore downstream. But everything on a path, I will be able to.
How much flexibility do you have in this intervention?
Oh, so this is perfect. Yes, so I will talk about that, and I think that's the main question nowadays, is now that we have this whole theory is experimental design, and this is also the main question that people ask me. So can we actually develop methods for experimental design of interventions, because there is full flexibility. So there is nowadays libraries for any gene, so you choose whatever you want and it will be done. And it doesn't cost more to do, this gene versus that gene, because there is a whole library of viruses. So this is done by viruses. So there's a virus that can do a knockout, and so you just put these viruses onto the cells and they will do the knockout. Yes, so we should talk about experimental design, and I'll have only one paper that we just started with experimental design at the end. But I think that's the biggest question now. Yeah. Yes? Oh, so, okay, so there's a whole story on how do you use neural network for learning these causal graphs, and they do also use things like graph neural networks, but they can do such small graphs. I mean, yeah, so it has not been successful yet in terms of gene regulatory networks, just because the graphs they can do are like 10 nodes or something. Yeah. But I think hopefully at some point, yes, I think it should go there. Also, you can use neural network, these nonlinear relationships will be represented by neural network, etc. So yes, definitely going in that direction. But I think it's important to first know what you can actually identify before just running a neural network on it.
What is the order of interventions play a role at all? If I change S1 first and then S2 first?
In this case, not. If I'm thinking about these, well, yeah. So usually, okay. So there is a whole other. So I'm thinking about biology now. So in biology, no, because whenever I want to get data, I need to kill the cell, right? So I cannot do one intervention, measure it, and do another intervention on top of it, because the cell is already dead. In order for me to measure its gene expression profile, I need to kill the cell. So all I can do is do both interventions at the same time. But yes, there might be other experiments where it matters. And actually, we're now looking at directed differentiation or something where I want to push the cell in this direction. I have a population of cells, right? I'll take out some and measure them, and the others I'll have to continue pushing them. So I think it will matter, but we don't know yet how much it matters. The assumptions are always it doesn't. Yeah. Okay. Okay, so then now that we know what can be identified, let's talk about algorithms. Okay, so in terms of algorithms, so in this paper here by Hauser and Bielmann for hard interventions, they also had a proposed algorithm of how to actually identify these interventional equivalence classes. Now we proved that they conjecture that it's consistent. We proved that it's not consistent. So even if you have an infinite amount of data, it's not going to give you the correct equivalence class, and also you cannot fix it, basically. Okay, so they took an algorithm that was already available in the observational setting and tried to change it to interventions, and one can show that that just cannot work. And so we had to start rethinking. I'll tell you a bit about how it works, but we had to start rethinking about algorithms in general, right? So because the algorithms that were there in the observational setting, you cannot make them interventional in a simple manner, at least. In this way, how it was done here, it can definitely not be done. And so what I want to do is tell you a bit about the classes of algorithms that were there in the observational setting, and then tell you about, because we have to come up with a new kind of class of algorithms for the observational setting that can then be turned into an interventional algorithm that actually is consistent, provably consistent in that setting. Okay, so this is a bit of a detour into older stuff, right? So these are the classes of algorithms, and it's kind of nice because it's so clean. The classes of algorithms that are around in the observational setting. So there are really two classes of algorithms. One is constraint-based. Here, the main algorithm is this PC algorithm. So constraint-based, how does it work? So you have data, and from the data, you learn constraints, which are conditional independence relations. Okay, so from the data, I learn conditional independence relations. And now what do? I try to find a causal graph that satisfies as many of these conditional independence relations as possible. Okay, now, this is usually fast. Also, you can actually show that these kinds of algorithms are consistent. So as the sample size goes to infinity, they will output the correct graph. But now in the finite sample regime, so what happens, and this is already clear, is they require huge sample sizes in order to do anything that is reasonable. Why is that? Well, because if I make any mistake in the constraints, I'm going to pay a huge price downstream, right? Because I'm trying to find these causal structures that satisfy as many of the constraints as possible. possible. So you might be, if I have too many constraints in particular, I'm going to remove edges, and then once you remove a wrong edge, you're going to make all kinds of mistakes in terms of orienting edges afterwards. Okay, so this you'll see when you compare the other class of algorithm, which are score-based to constraint-based, with the same sample size, score-based methods will always do much, much better. Okay, yeah.
Why do you call it fast? Isn't it trivially exponential time?
Everything is NP-hard. I'm just comparing these types of algorithms against each other. And let me talk about why fast when I'm talking about the score-based approach and what the problems are there. So everything is NP-hard. In the worst case, no, it will be very slow. And that's when you have a complete graph. You just have to search over all possible orderings. But hopefully, in general, the graphs are not complete. So otherwise, there's anyways no interesting causal structure. Okay, so the other class of algorithms are score-based. So here, usually you take some score, like a penalized likelihood score, a BIC. And so what you do is, so now you want to search over all causal structures and just maximize the score. Okay, and so, well, the space of graphs is huge, so what you do is a greedy search on the space of graphs. And so the most, well-known one here is this GES, greedy equivalent search, which is actually a very interesting algorithm, because usually for greedy searches, it's hard to prove that they're consistent, right, that they don't get stuck. But actually here you can prove that this one doesn't get stuck. So what it does is it starts in the empty graph, it adds edges until it cannot improve the score, it removes edges until it cannot improve the score, and then it stops. So just one forward, one backward search, and it's actually consistent as the sample size goes to infinity. Okay? So this is very interesting that it is consistent, first of all. And now I will talk a bit about the advantages and disadvantages. So statistically, this usually does much better than this other algorithm because, even if you have some mistakes in terms of conditional independence relations, because it's greedy, it doesn't matter, right? I can make a mistake in some step, and hopefully, as long as I can still move forward to some better graph, I'll still be fine. Okay, so that's why this usually does better statistically. Now I'll talk about what the problems are in terms of computation times. Okay, so let me just, and this is actually the algorithm which Hauser and Bühlmann took and tried to make interventional, which is provably not consistent and cannot be changed into a consistent algorithm.
So right now to see is the relationship between those partial relationship or the strength of the relationship?
Correct, yes. Here you're just trying to learn the graph. Yeah. And then once you have the graph, you can do all kinds of stuff afterwards. Okay, so just a bit about, and I already hinted at the advantages and disadvantages of each one of them. So constraint-based, and let me actually just put out everything. And just to give you intuition, it's exactly this. It's like the problem is that you often make mistakes in the constraint. And so here comes this faithfulness assumption, which is just the assumption that says that, well, whenever there is an edge in the graph, I should not be learning that they are independent given anything, right? Because otherwise I'm going to remove the edge and I'm going to make a huge amount of mistakes downstream. That's what the faithfulness assumption is, okay? Faithfulness assumption says that the distribution is faithful to the graph, so there are no additional conditional independence relations. Now it might seem like a very harmless assumption, and that's why, it was proposed and never thought about. But what does it really mean? If I give you an example, you have like some medicine that has a positive effect on the lung functioning, and it's so strong that it has a negative effect on the immune system. But of course, a well-working immune system has a positive effect on the lung function. Now, it can be that this positive effect and this negative effect cancel each other out. Okay, and that's exactly a faithfulness violation, right? Because in that case, you would have the medicine is independent of lung functioning, and you would remove the edge, and in particular, then you will get actually wrong edges pointing downward. So even the causal structure will be wrong. And so this is problematic, right? I can even remove weak edges, and then I get all kinds of wrong causal relationships afterwards. So faithfulness violations are really problematic. And, yes, you can say, well, this happens with probability 0, right? It has to be that these causal effects exactly cancel each other out. Well, but, you have finite sample sizes. So I'm going to do a hypothesis test, and I'm going to say, well, if this, partial correlation is smaller than, I don't know, some value, I'm just going to assume that they're independent. Well, so that means, so now this is in fact a 3-no case. In the 3-no case, you have 6 possible independencies, right? x1 independent of x2, x1 independent of x2 given x3, etc. So there are 6 possible independence relations. If the true graph is that one, then I'm not allowed to be close to any one of them. Okay, and in fact, you know, if I have a 3-node graph, I have 3 variables. I can even plot these conditional independence relations in the Gaussian setting. So these are it, okay? So what it means to be faithful is that you have to be bounded away from all of these hypersurfaces. Because if I come close to any one of them, I'm going to make a mistake. And so you already see that this is problematic, right? So if I have a finite sample size, so in fact, how much you have to be bounded away is, you know, some square root of log p divided by n. So, and we can just prove that, you know, if just you have enough, I mean, you just need such a huge sample size because otherwise, if you just fatten up these hypersurfaces, you're going to fill up the whole space. So there is no faithful distribution. Just doesn't exist. Okay, so this already tells you that, you know, this is a very strong assumption, right, that you need in the constraint-based setting. Okay, so that explains why statistically these algorithms usually don't do as well. Now, what about the other score-based approaches? So in score-based approaches, so actually, so they put in quite a bit of effort in this GS algorithm that, you know, since in the observational setting you can anyways not identify the full causal structure, but only identify the Markov equivalence class, their search is even just on Markov equivalence classes, okay, instead of on causal structures, so to cut down the computational costs. Now, the question is that worth it? And, you know, how large are these spaces where you're actually doing greedy search? So it's, in fact, it's not known. So, okay, so there is a formula for DAGs, right? How many DAGs are there on, say, P nodes? So there is a formula. What is interesting is that there is still no formula known for how many Markov equivalence classes are there on P nodes. Okay, although there is a very nice graphical characterization. Okay, there's still no formula known. So all there is these enumerations of here up to 10 nodes. I think we did up to 13 or something. But so here you see that this really explodes very quickly, okay? So you have about 10 to the 19, I think this is, equivalence classes on 10 nodes. So this space is big where you're doing greedy search. Okay. Now, what I think is more interesting, I mean, this is already a nice conjecture, and one should actually prove something like this. But I think what is more important to prove is these kinds of numbers here, which they also reported in this really nice paper by Gillespie and Perlman, where they look at the number of equivalence classes divided by the total number of DAGs. So this is to ask what is the expected size of a Markov equivalence class, right? So you know, in this GS algorithm, they put in so much effort to make a search over equivalence classes instead of over DAGs, hoping that this number would be big, right? Because otherwise, it doesn't really matter. So here, what it seems, and this would be really, I think, important to prove, is that this number actually converges to something like a quarter. So in average, an equivalence class has about 4 members. Okay, so you know, if we go back to this little picture, you should think that even for very large graphs, the picture is kind of like this, where there will be a huge class, namely the complete one is always of size p factorial, but there will be so many small ones. Okay, and usually, you know, you don't really care about this big one here. So I think that's a really important conjecture to prove, which, you know, is still out there. And maybe even this one, in terms of interventions, is really interesting. Namely, the question of if we look at all Markov equivalence classes, and here of all Markov equivalence classes of size 1. Okay, so this means when in the observational setting, you're actually able to identify the causal graph completely, right? So this also seems to converge to something like a quarter. Okay, so that means in a quarter of the cases, you don't even need any interventions. You're going to be able to identify the graph. And in all other cases, you actually need very few interventions. Right? This is like the experimental design question, but you need very few interventions in order to be able to identify the graph, but it matters a lot which ones. Okay, so it's probably important to start off with observational data and then try to figure out which interventions do I need in order to actually nail this down. But I think these are really interesting kind of conjectures that are super important for causality and that we still haven't proven. Okay, so we looked at them recently for random graph models, so Erdős-Renyi graph models. So there we were able to prove something about these. But, you know, in full generality, I think, you know, someone who knows some combinatorics should be able to actually prove these kinds of results. Right? And I think it would be really important for causality. Okay, so that's the problem. When you do greedy search on this space, it's just a big subspace. So fast and slow is compared to this, okay? Okay, so these are types of algorithms that have been around in the observational setting. And now the question is, how do we get something that is actually consistent in the interventional setting? Okay, so when should I stop? Should I leave time for questions or? Okay, so I just continue like this. Okay. Okay, so now let me actually propose an algorithm first in the observational setting that we can actually make consistent in the interventional setting. And it will solve some of the problems that we just saw about, slow and fast and not being a constraint-based algorithm and being a score-based algorithm, etc. Okay, so, and the idea is super, super simple. Okay, so instead of doing a search over graphs or equivalence classes, well, if you want to learn a DAG or an equivalence class of DAGs, what identifies a DAG is just a permutation, right? An ordering and an undirected graph. Because if I give you the undirected graph and I tell you, say this is my undirected graph, and I tell you that 1 is smaller than 2 is smaller than 3 in the ordering, well, then I know that, my edges have to be pointed in this direction, right? Because it is a DAG, so I cannot have any directed cycles. So the ordering will identify the DAG as long as I have a skeleton. Okay, so now, we'll talk about undirected graphs, etc. But believe me that there is, for example, Pearl in 1988, he already showed that if you give me the ordering, it's actually pretty easy to identify the undirected graph corresponding to that ordering. And this is known as this minimal IMAP, and it can be computed, etc. And I have here a formula. But if I give you the ordering, then basically learning the undirected graph corresponding to that ordering is as easy as standardly learning an undirected graph. Okay, and it can be done, and we know exactly which conditional independence relations have to be tested. So that means that really what is hard about causality is just learning the permutation, learning the ordering. Okay, because once I give you the ordering, you learn the corresponding undirected graph, and you continue on. So that makes sense to, instead of do a greedy search over all Markov equivalence classes, well, let me just do greedy search over the space of permutations. And the space of permutations, is much smaller. Now, again, we don't know how large the space of Markov equivalence classes is. At least for 10 nodes, it's 10 to the 18, so this seems much smaller. So it seems like a much smaller space in order to do a greedy search on. Okay, and this is really what matters, right? So I'll propose to be doing a greedy search on the space of permutations. So what do I need to do in order to do greedy search? I need to give you a score function, and I need to tell you how to do greedy search. Okay, so we want it to be non-parametric, right? So not like BIC or something like that, where I need to make a Gaussian assumption usually. So I want it to be completely non-parametric. So my score function is just going to be the number of edges. So the simplest possible score function. Okay, so you are in some permutation. You learn the corresponding undirected graph, and hence DAG. And I just count the number of edges. That's my score, and I just want to minimize that. Okay? Now, that's my score, and the other thing I need to tell you is how do I do greedy search. Now, the space of permutations is very nice. It's actually, if you take the convex hull, it's a nice polytope where each one of the vertices is actually a permutation. And so, well, if I want to do greedy search, so this is known as a permutahedron, well, I'll just go along the edges. Right? And so this has been studied a lot. So here, what are edges? So my neighbors in the space of permutations are just going to be given by neighboring transpositions. So for example, if you're here, first of all, you see it's very regular, right? Everywhere there are 3 edges adjacent to it. Namely, I can permute the first 2 guys, I get to 1, 3, 4, 2 in this case. I can permute the next 2 guys, I get to 3, 4, 1, 2. Or the last 2 guys, I get to 3, 1, 2, 4. Okay, so I look at my neighbors, I look at their graphs, I count how many edges there are, and I just walk there if there are, no more edges than there was. Okay, simplest algorithm. And you can prove that this is in fact not going to get stuck. Okay, and so the easier proof is to show that the sparsest one is actually the true one. That's the easier proof, and then the hard proof is to show that you will never get stuck. So you can start anywhere and you won't get stuck. Sparsity? Just the number of edges. So don't care about directions, just the number of edges.
If I have a permutation and I fill in on the edges, is that also?
Yeah, okay, so for every permutation, to every permutation, I can get the corresponding graph, which is this minimal IMAP, in some way. And so in this graph now, you just count the number of edges. Okay, so this defines, so all I'm doing is I'm testing, so to know if I have this edge, I'm going to test is this guy independent of this guy given this guy. So for every permutation, if I maybe here change the permutation to 2 is smaller than 1 is smaller than 3, right? In this direction, it might be that I'm actually losing an edge. So it might be that my graph then just looks like this. And hence, in that case, I would actually move here. Okay, so to every permutation, I have an associated undirected graph, and I'm counting the number of edges in there. And actually, I have an example. Sorry, I should have done this. So here I'm telling you I'm doing greedy search on the space of permutations, but you know, multiple permutations can have exactly the same graph, right? So in particular, if I'm in this guy, 1 is smaller than 2 is smaller than 3, then obviously in this permutation I have exactly the same graph. So I don't want to search over a space where I already know that the graphs are the same, so I'm going to glue these together. So whenever you have permutations that have exactly the same graph, I'll just glue them together. We can actually prove that this is, again, a polytope. That's not trivial, right? So if I take a polytope and I just glue together edges, in general, you don't get a polytope out. What is really nice is here you actually get a polytope out. Okay, and now here you see it, right? So I'm in some ordering. I'm here in this guy. This is my graph corresponding to it. I look at my neighbors. And here I show you how they look like. And this guy, for example, would be sparser, or that guy, so you just move there.
Identification between permutations is done given the independence relationships of the data.
Yeah, so this is actually a good question, right? So it would be bad if I first have to compute this polytope in order to actually then do causal inference. So you don't have to, because I can identify who are my neighbors without knowing the polytope. So I know who my neighbors are just by looking at this graph. Namely, I know which edge I can possibly flip. So
again depends on knowing the independence relationships of the data.
Correct, yes. So you learn from the data. All I'm going to test is like here, for example, I will know which ones I have to test in addition. I'm not always testing all of them. I'll just test the ones that I can possibly remove. Yeah. Okay, so this is in the fully observational setting. So it's now just greedy search over the space of permutations. And now, in fact, this algorithm can just be made very, very simply into an interventional algorithm that is provably consistent. And so let me just go through how you would do this. Oh, sorry, perfect is the same as hard. So how would I do this for hard interventions? So let's just think about it for hard interventions, okay? So I'm doing this greedy search over the space of permutations. So I'm going to do just the simplest possible thing. Namely, if I'm in this permutation, I have a corresponding graph. Okay, I look at my neighbors. And so if I look at this neighbor here, right, I'll make some edges now point. I'll be turning around some of the edges. Like here in this case, for example, I turned around the edge, right? And so if I have a hard intervention, I already know that there cannot be any